And this diagram explains you the different routes of administering your drugs. So the enteral drugs will be taken orally. It's going to go into your digestive system and it will be absorbed through there. Okay, those are the enteral drugs. And then parenteral drugs on the other side will be administered through injections. Okay, so they'll be absorbed through your skin, muscle, or different tissues in your body. And then in terms of injection, the needles can be placed in different parts of your body. It depends on how deep it's penetrating your body. So it could be intramuscular. So it's like via vaccination will be done on your buttocks. Okay, so that's where it's going to be injected, your drug. And then subcutaneous is a little less penetration than muscular injection, but it's normally given to the fat tissues in your body. And then intravenous is through your blood vessel. Okay, and then intradermis is right below your epidermis. Okay, so different ways of giving injection. So again, parenteral drugs. So the word, it comes from two Greek words. Para means avoid, okay, or skipping. Enteral, okay, as I said, is the bodily canal is what enteral means. So you're avoiding the bodily canal. So it's basically injection. That's what it means, parenteral. So injection or injectable drug is what it means. So normally parenteral drugs are made of peptide, okay, or proteins or some biological molecules. So if they are injected orally, they may be denatured or damaged. That's why they have to be given through injection. Okay, and so like I said, mostly made up of biological molecules, just like insulin. So insulin is not given orally. Normally it's given through injection. Okay, since those drugs are given through injection, so they have to be really clean, free of contamination. That's part of the requirements. So those should be sterile drug. Sterile means free of any contaminants, right? So products should be introduced in a manner that circumvents the body's most protective barriers. So your epidermis is the most protective barriers. So epidermis, and then the digestive system is also made out of your epidermis. So since you're bypassing those, so your drug should be really clean and safe. Okay, so they should be sterile, free of contaminants. So that's why when these drugs are manufactured, they should be manufactured aseptically. So that's part of the CGMP requirement. So understand that. And then now let's talk about the drug product development cycle. Probably you've seen this in your previous classes like BIOT100 or 102. We talked about it so many times. So it's hopefully a review for you. So when a drug is developed, it takes a long time, in average about 12.5 years of time, and the investment about more than $1 billion, $1.1 billion. Okay, so that's how you bring your idea concept from bench scale to all the way to the market. It's a long time and lots of money, right? And also you can get your idea patented, right? In the US, normally the patent is good only about 20 years. So 20 minus 12.5 years, you have about 7.5 years to get your money back. Okay, so that's why drug prices are high. Okay, and then developmental processes, the stages, of course, your bench idea. Okay, idea, and then preclinical testing, and see if your drug molecule is good in terms of its pharmaco efficacy. Okay, you're going to test it on your maybe in vitro cell lines or animal models like mouse or chimpanzees. Okay, and see if it's effective. And once all the data is good, then you will develop a plan for your clinical trials and file IND, new investigational drug. Okay, filing with the FDA. Okay, and then after about 30 days, you can start your clinical trials, phase 1, 2, 3. Right, and after phase 3, of course, you can apply for a new drug approval submission. Okay, so clinical trials, you know what's happening at each stage. Phase 1, a small group of human volunteers, mostly healthy, without any medical symptoms or conditions. At that stage, you're going to test the safety of your drug normally, see if there's any side effects, bad side effect. So if your drug is safe, of course, then you're going to move on to the phase 2 trial, and you can test your drug on a lot more people. And at this stage, you're going to apply your drug to the people with medical conditions, the patients. Okay, so patients are recruited, about 100, 300 patient volunteers, and you're going to test for the safety as well as the efficacy of the drug. So efficacy is tested starting from the phase 2. Okay, and then if the data comes out to be positive, then you can move on to the phase 3, which is much larger scale, maybe 10 times larger scale, 1,000 to 3,000 patient volunteers again. You continue testing on the safety and efficacy of the drug, and at the end you can file an NDA. Okay, so we talked about the corona vaccine development. The drugs that are approved have gone through this process. Moderna drug or Pfizer drugs, they have got their approval through going through their phase 1, 2, and 3 trials, right? And after that, FDA will take their time to review your filing. Okay, if everything is successful scientifically, then they will give you approval. Only then you can put your drug on the market. And then it's not the end. The phase 4 is after putting your drug on the market. You're going to have to place your post-market surveillance for any kind of side effect, any kind of issues among the patients. Okay, you'll probably have to set up a hotline, 1-800 number, or website. If there's a problem, they will contact you for that. Okay, so that's that. So phase 4 right there. So it's a long process, of course, and your drug manufacturing will start, okay, right here. For your clinical trial, you have to have a drug that is safely manufactured. So CGMP starts during your clinical trials, okay, and for your marketing, of course, your GMP manufacturing scale will go up for commercial size. So that's the process. So all these things, the analytical methods are developed prior to the final dosage form, which helps validate efforts prior to IND filing. So before you start your clinical trials, you have to have your method of manufacturing, method of testing, method of dosaging your drug, formulating your drug should be kind of developed. That's what it means. Okay, and then so clinical trials takes many years. Phase 3 will be the longest. Okay, it involves many large patients, so it takes a long time. And final formulation package process should be locked in before or during the phase 3. So before your NDA approval, it means you have to have your final formulation, packaging method, and materials, and everything should be all developed. That's what it means. So before you submit your NDA, you have to have it in your form. Final production specifications are finalized during the phase 3 batch production. Okay, prior to the submission of NDA again. Okay, and then process validation and batch production should be initiated prior to submission of NDA. So normally, NDA is the kind of final submission, so everything should be locked in. That's what it means. So understand that. So now, when you manufacture a biological drug, of course, there are two main stages. The first stage is drug substance manufacturing. So you probably learned about upstream processing, downstream processing, where you're going to have your genetically engineered cells that are producing your API molecule, maybe proteins. Okay, and so from 1 ml frozen ampule, you're going to grow and scale up all the way to maybe large size batch bioreactor, maybe 2,500 liter volume. Okay, and then you're going to have the cells express your protein, and then you're going to clarify them, and then you're going to purify them through various steps of downstream processes using a bunch of chromatographic columns, filtration methods, right? And once that's done, you're going to start formulating your drug by mixing, compounding your drug with the excipients. the chemicals that can stabilize your drug. Okay, so that's the beginning of the drug product manufacturing. So upstream, downstream steps are part of the drug substance manufacturing. Then after that, the drug product manufacturing will follow. So that is the focus of this class. So formulation, the first step, and then aseptic filling. So formulated drugs should be filled in the primary containers, maybe a drug vials, or maybe syringes, or ampoules, whatever that you're going to use as the primary container. Okay, and those primary containers will be kept and sealed. Okay, and maybe cramped, or maybe syringes will be sealed as well. And sometimes the vials may be lyophilized, meaning what? Freeze drying to remove the water. So if you remove water, there will be less chance of hydrolyzation of your drug molecule. So it'll stabilize your drug for longer time, for longer storage on the shelf life. And then you're going to seal it, crimp it, and then package it, secondary containers, and all those things, and distribute it. So all these steps are part of the drug product manufacturing. So we're going to learn about all these steps. That's the purpose of this class. Okay. So review the information on biologic and their manufacturing requirement from the website below. So if you go to the website made by the company called Genentech, you'll be able to learn a lot of information about drug manufacturing process. So visit there. This manufacturing, they have really nice educational information on this website. Into the future, giving advice, investing diversity, how it's made, upstream, downstream processes, and all those things. Beauty of manufacturing. So spend some of your time and go through a lot of these pages. These are really helpful. Okay. All right, and then so yeah, that's the end. So I have these 4 questions as your assignment questions out of this lecture. So I want you to download this question if you go to the assignment section. So I'm going to post these questions there. So I'm going to combine the questions from the lectures 1 and 2, and that'll be your first assignment, 10 points. So download the document on your Word, and then type up your answers to these questions and save it as a file and upload it back to the assignment section, and you will get 10 points for that assignment. So you will get this at the end of every lecture, every week. So these will be part of your assessment for the course. All right. Okay, so the first question is, what are biologics and how are they different from small molecules? We already talked about it. Small molecules are chemical-based small drugs, but biological molecules are larger drugs because they are made out of biological molecules, much larger. Okay, and enteral versus parenteral drugs. Okay, enteral is taken orally, going through your digestive tract. Parenteral, injection, because they are sensitive drugs that can be denatured if they are taken orally. Parenteral or sterile drugs. Why should the parenteral drug be manufactured under CGMP? Because parenteral drugs given through injection, they are bypassing the natural defensive system of your body, so they have to be free of contamination. They have to be sterile, right? And then during what stage of clinical trials or drug product development is CGMP engaged? Good current manufacturing practice. So when does it start? Even before the clinical trials, right? Okay, so yeah, write down your answers and upload it. All right, thank you.